It is my pleasure to introduce to you Nathan Gamarra. Nathan obtained his Bachelor of Science at UC Irvine, where he already got a lot of research experience in, and he was awarded the Dean's Award for Excellence in Research. He then got an NSF Graduate Research Fellowship and started grad school at UCSF, where he joined the lab of Narlikar. There he's now done fantastic work on ATP-dependent remodelers, specifically SNF2h, and we will learn a lot more about that today. So Nathan is about to obtain his PhD very soon and will then start a postdoc at Genentech. So I'm sure he'll have a lot of exciting stuff to talk about in the future as well. But for now, we'd love to hear what you've been up to, Nathan. Go ahead.
Awesome. Thank you so much for giving me the opportunity to share my graduate work. Here we go. Today I'm going to tell you about a story about how a molecular machine uses sort of molecular motions within the nucleosome to achieve its ends. And so, so as we all can appreciate in this audience, the regulation of chromatin structure is very important. And it really occurs at the nucleosome level. And one of the major workhorses of regulating chromatin structure are the ATP-dependent chromatin remodeling enzymes. And these are large multi-subunit molecular machines that use the ATP hydrolysis to disrupt nucleosome structure in a variety of ways, kind of shown here. So I actually focused on one of these particular remodelers that Hanneke already mentioned, which is SNF2h, which is a member of these I-switch family of chromatin remodeling enzymes. And these enzymes use ATP hydrolysis primarily to slide nucleosomes along DNA, so move the histone octamer with respect to DNA. And this can be used for a variety of different means, one of which shown here is in regulating sort of access to the promoter for pre-initiation complexes. And so primarily what I-switch complexes do is they slide nucleosomes away from barriers. So any sort of barrier, such as, say, the pre-initiation complex, SNF2h will prefer to slide nucleosomes away from said barrier. And so we can actually model this process in vitro and learn with great depth about how remodelers execute their functions simply by purifying the ATP hydrolyzing subunit of the I-switch complex and giving it a nucleosome and a single nucleosome. And in sort of an ATP-dependent way, SNF2h will move the nucleosome away from an end, which to SNF2h is essentially a barrier, and will move the nucleosome towards the center of a piece of DNA. So in this case, essentially what SNF2h is doing is equalizing the lengths of DNA flanking on either side. And we monitor this reaction using this native gel-based assay. So nucleosomes, when reconstituted, can be run on a native gel, and they have a weird property where if they're less centrally positioned, they migrate faster through the gel than if they are more centrally positioned. And so we can monitor the reaction over time and get nice quantitative measurements of remodeling. And so a really basic question is, how is SNF2h able to accomplish this rather, complex reaction of mobilizing the nucleosome to a very, somewhat specific location? And so it turns out that this single protein can accomplish this because it's actually made up of several parts that all work in very complex ways. So if you look at the domain architecture of SNF2h, it has a ATP hydrolyzing subunit, which also does the main work of mobilizing the nucleosome. And additionally, it has other domains such as this HSS domain, which is actually the physical element that senses flanking DNA. And these two regulatory motifs that have been somewhat poorly characterized, this N-terminal and C-terminal autoinhibitory domain called AutoN and neg-C. So how can all these different components work on the nucleosome? Work from many labs, including Felix's lab, who will be speaking after me, have really shed a lot of light on how this process can work. And so how we really think this functions, how SNF2h functions is it initially binds the nucleosome with its ATPase domain somewhere internal to the nucleosome core, or internal to the nucleosome. In this state, it can then sense the length of flanking DNA by having its C-terminal HSS domain binding flanking DNA. After binding flanking DNA, this stimulates ATP hydrolysis and shifts the enzyme from this auto-inhibited state where it's just looking for where to slide into a state where it can actively slide the nucleosome. And this is really the major question, I think, that I'm kind of interested in is, what keeps this, what regulates this switch into an active state, and what keeps it into this active state when it's sliding. One element of this is this histone H4 tail, which can bind the ATPase domain and stabilize the active state, but there are potentially other unrecognized features of the nucleosome or the remodeler that may be important in this. And a second question, which really complicates the picture, is we have found that actually two copies of SNF2h, two SNF2h proteins can bind to the nucleosome at the same time. And this is actually the most efficient way that the nucleosome is slid. And this is simply because the two protomers can communicate with each other to decide which direction to slide. And we think that this really happens at this auto-inhibited step where both protomers can take turns sensing the length of flanking DNA and ultimately decide who wins the slide. But really, what prevents the opposing protomer from competing and ruining the active protomer's ability to slide is a major outstanding question, and something that I'll also address. So starting with the first question, what keeps the sliding protomer active? If one feature of the nucleosome that's been really appreciated for a while is the acidic patch, which is, this audience will know really well, is a region of high negative charge in histones H2A and H2B that a lot of proteins use to bind the nucleosome. So I am really interested in whether this could be playing a role in SNF2h mediated remodeling. And indeed, if I assemble nucleosomes where these residues are mutated to alanines, we see remodeling is slowed quite a bit. So a reaction that's done essentially within 5 minutes takes multiple hours to not even be fully complete. This could be due to a lot of reasons. One reason could simply be that SNF2h is unable to bind the nucleosome like many other proteins. But it could also be due to some sort of ability to switch on the activity of the enzyme. And so to test this, I simply varied the concentration of SNF2h and measured these two parameters, Km, which reflects binding, and Kmax, which reflects activity. And what you can essentially see here is that a reaction that in the sliding reaction, while there is still a 5-fold defect in Km, which reflects binding, there is a much larger defect in Kmax, the actual activity of the enzyme. So this suggests the acidic patch is really being used potentially to switch on the activity of the remodeler. So this returning to this schematic, what is actually happening that is causing the acidic patch to enable this active state. So to do this, you need a way to be able to selectively enrich for the inactive and active state, and we can actually do this by giving SNF2h nucleotide analogues. So ADP will mimic this auto-inhibited state where you're binding flanking DNA, and ADP beryllium fluoride will mimic sort of an activated state. And really a major question that we had is, what is happening at the acidic patch? And so we perform cross-linking mass spec in both of these two states. And what you can see is more red indicates more interactions with the active state, and more blue indicates more interactions with the inactive or auto-inhibited state. This was done in collaboration with Mike Trinka's lab at UCSF. And we got data for all possible pairwise protein-protein interactions. But if we just look at the acidic patch, what we can see is interactions across the whole protein seem to increase in the active state. which was pretty interesting, and we can get into why that might be. But one thing that really stood out to me was these two relatively uncharacterized autoinhibitory domains seem to increase their interaction in the active state. So I just wondered, what would happen if I mutated these different autoinhibitory domains and measured remodeling on a mutant nucleosome. So you can see on a normal SNF2h, the reaction is quite slow. But if I mutate AutoN, in this case, just by replacing two residues with alanines, the reaction is quite a bit faster. So the reaction now is done within a similar time scale as a normal nucleosome. And this is also seen in the case of neg-C, where in this case, I simply replace the whole motif with a flexible tether. So in other words, as shown here, normally something that is dependent for activity around 200-fold, if we mutate these autoinhibitory domains, this SNF2h becomes only 8-fold or even 2-fold dependent on the acidic patch for activity. So this really suggests the acidic patch's main role is in relieving autoinhibition. So this is just a physical model for what I just said. There's an autoinhibited state, and somehow interactions with the acidic patch, in addition to interactions with the H4 tail, allow the active protomer to be stabilized and slide the nucleosome. But now, what happens in the context of the dimer, where we have two protomers bound to the nucleosome? And so, to get at this question, I need to actually introduce a key aspect of nucleosome structure that's really emerging in the field today. So, many people sort of typically think of the nucleosome as this rock where it exists in a single conformation once the DNA is wrapped around the histone proteins. But it turns out work from our lab, as well as the Luger and Halleck labs, have suggested that the nucleosome is actually way more plastic than we anticipated, and that the conformation of the histone core can change or flex in response to different cues or just naturally. And indeed, our lab has shown that that SNF2h, when it binds the nucleosome, may induce conformational changes within the histone octamer that are important for sliding. And so what we really want to get, though, was a direct picture of these conformational rearrangements. And so to do this, we obtained cryo-EM reconstructions of SNF2h bound to the nucleosome. And a key aspect of cryo-EM is that anything that is rather dynamic, that has multiple conformations, is sort of lost in the reconstruction. So what you can see here is we can very clearly resolve in these different 3D reconstructions SNF2h bound to the nucleosome, its ATPase domain bound to the nucleosome, but its other domains are invisible. And this is simply just because they're so dynamic that they average out in cryo-EM. So averaging out is kind of a proxy for dynamics. And so what happens at the acidic patch? Well, it turns out that if we look at a complex of SNF2h bound to the nucleosome, which we think is the protomer that is sliding the nucleosome, if we compare the two faces of the nucleosome at the acidic patch, we see that the face that SNF2h is bound on has much more resolved EM density than the opposite face. And so what we think perhaps is happening is that SNF2h is inducing conformational rearrangements or dynamics in the acidic patch of the opposite face of the nucleosome. And at this point, this is a little bit speculative, and I'm working on testing this hypothesis. But I'm giving you a picture for what I think is going on. So this is just a schematic of that I've shown earlier, but now with the nucleosome rotated to see it from the side. And so what we think happens is that when SNF2h rearranges into its active state, it actually induces asymmetric sort of dynamics within the nucleosome, specifically in the acidic patch on the opposite face. And these dynamics may prevent the ability of the opposing protomer from competing with SNF2h to slide the nucleosome. And so this, in some ways, prevents the tug of war that I was sort of alluding to earlier. And so what you can see essentially is that all of these features of SNF2h and of the nucleosome are important, in terms of creating a very specific structure that's SNF2h uses to slide. Now, there's still many open questions. In particular, how does of the more relevant in vivo complex known as ACF, which is composed of SNF2H and this large accessory subunit called ACF1, actually functions on the nucleosome? I'm also really interested in this concept of how does SNF2H interface with other proteins that use the acidic patch to slide or otherwise modify or bind to the nucleosome. And I imagine that perhaps some of these principles just within the SNF2H dimer may be important for other protein complexes negotiating access to nucleosomes. And really, I think an important question is, what is the role of these conformational dynamics that we detect in vitro, in vivo? And, and one aspect also is that the nucleosome is a symmetric structure, but there's several lines of evidence that it can adopt sort of asymmetric structures. So especially here in terms of conformational rearrangements, but modifications, etc. So what role does this asymmetry play in remodeling and other chromatin proteins? And I want to thank the lab for all of their help and support, as well as Yifan Cheng's lab, who I collaborated with to do CryoEM, the Burlingame lab for cross-linking mass spec, Song Tan's lab for help generating the acidic patch mutant nucleosomes, and Greg Bowman's lab for a lot of very helpful discussions and experimental help as well. Thank you, and any questions would be appreciated.
Great, thank you, Nathan. That was fantastic. Okay, so just as a reminder, people can type their questions in the Q&A box, or they can raise their hand while people are working on that. I know that Christine already has 2 questions, but I'll ask her to stick to one. Do you want to go ahead, Christine?
Thanks. Yeah, so I guess which question is the better one? I don't know. Do you want to answer an H2AZ question or a ubiquitin question?
Both are great.
Okay, I'm going to ask you, basically they're both similar. So have you looked at either from the cross-linking mass spec or activity of SNF2H with either monoubiquitinated nucleosomes, since that H2BK120 is so close to the acidic patch? Does it do anything to activity on SNF2H, or the extended acidic patch on H2AZ? I'm interested if that has any impact on the SNF2H activity.
Those are great questions. And so H2AZ has actually been well known to actually enhance the activity of SNF2H slightly. So the extended acidic patch seems to influence its activity. As for ubiquitin, I don't believe I've seen anyone. I know that Tom Muir's group has done extensive looking at different modifications and how they influence I-switch remodeling, and I can't remember if they tried ubiquitin. I would think they would, I'm not 100% sure, but I'd imagine it would influence, because as you said, the modification is quite close to the acidic patch.
Cool, thanks.
Next, we'll have a question from Alexandra Skreiner, who I've just allowed to unmute herself. Want to go ahead?
So I have a quick question. Did you try to do any FRET to try to see the interaction, like between the region that is very flexible and you are not able to see it using CryoEM?
Yes, it's quite challenging for a lot of reasons, but I did attempt to, and the assay just never, I never got it up and running fully. But that would be a great sort of future direction, I think, is to really try to nail down some of these dynamic interactions. And I think FRET is a key, would be a key sort of method to do that.
Yeah, well, good luck.
Okay, great. Then we have a question typed out by Ilana Nodelman. And she says, nice talk. Did the more dynamic side in the CryoEM structure correlate with the TA poor side of the 601 sequence?
That is a really good question. So I always forget. I believe the TA and Ilana would know this really well because of their work. So we never flipped the 601 sequence, so I don't think we could ever really tell for sure whether the sequence really influenced the dynamics. But in our structure, it's the side opposite the SNF2h protomer binding, which I believe is the TA rich side of 601. We can talk about this later, but in other words, we can't know for sure whether sequence is driving these dynamics or whether it's just SNF2h binding.
Right. Okay, and I have a final question that is from Janet Young. And she asks, in vivo, where does SNF2h reposition the nucleosome to? That is, when it's not working on short DNA fragments.
I see. So in vivo, SNF2h functions at a lot of different places. As I sort of showed in the schematic earlier, it plays a key role in positioning the plus and minus one of the nucleosome at transcription start sites. It also plays key roles in repositioning nucleosomes to enable DNA repair. And it also seems to play a very, very key role in setting up even-spaced nucleosomes near CDCF cohesin binding sites. So it functions at a lot of different places within the genome, and yeah, and sort of its repositioning activity. Really, it's not fully clear exactly how these specific repositioning leads to very specific biological outcomes, and that's, I think, a kind of a frontier that I'm really interested in actually hopefully at Genentech to kind of address.
That sounds like a perfect future outlook to end off on. Thank you so much, Nathan.